Hello everyone, and thanks for joining BioTeach on this video on food tests or biochemical food tests. This video will take you through the 4 biochemical tests you need to learn, as well as a couple of exam questions at the end. Biochemical tests are qualitative tests which can be used to detect the presence of molecules such as lipids, proteins, or carbohydrates. One of the advantages of these tests is that they're really quick and give a colour change result which can be easily observed, but they cannot be used directly to determine the absolute concentration or distinguishing between different types of molecules of the same type. So, for example, different sugars in a mixed solution. Let's first look at the test for starch. You may have had a chance to carry this out when you studied for your GCSEs. The iodine test is the test that allows us to check for the presence of starch. There are a couple of methods, but we usually use a dimple tray, like the one shown in the image on the bottom right. You would need to add a drop or two of the test solution on the spotting tile, and then add a drop of the iodine or potassium iodide solution. If starch is present, you would see a blue-black colour change occur, as it's shown in that photograph on the bottom right. The next test you need to know about is the Benedict's test, which is used to test for presence of sugars. There are 2 types of Benedict's test, one for reducing sugars and one for non-reducing sugars. A reducing sugar is a sugar that has a free aldehyde or ketone group that can act as a reducing agent. A non-reducing sugar does not have a free aldehyde or ketone, so it cannot act as a reducing agent. All monosaccharides are reducing sugars. The disaccharides of maltose and lactose are also reducing sugars, but sucrose is a non-reducing sugar. One of the key things to note about the Benedict's test is that the more reducing sugar there is present in a solution, the more the colour change occurs. The diagram on the bottom right shows you how to carry out a Benedict's test. A sample of your solution is added into a test tube and is mixed with an equal volume of Benedict solution. Benedict solution is blue in colour, and that is usually your starting point to monitor any colour change. The tube is placed into a water bath and heated. You would observe and record any colour changes. If there is a reducing sugar present in the solution, the colour would go from blue to green to yellow to orange and finally to brick red. Brick red denotes there is a high concentration of reducing sugar present. If the colour remains the same as the copper sulfate solution or the Benedict solution, so if it remains blue, it could mean that it's a negative result, that there is no sugar present, or it could mean that there is a non-reducing sugar present. So for a sugar such as sucrose, we need to carry out a couple of additional steps. We know sucrose is a disaccharide, so we would need to break it into its monosaccharide constituents. To do this, we would boil the solution with diluted hydrochloric acid and then cool it and neutralize it with hydrogen carbonate. Then we would carry out a Benedict's test by adding Benedict solution to your sample and putting it on a water bath and looking for a colour change to brick red. A positive test would mean that a non-reducing sugar was present, and a no colour change test would show that no sugars were present. I should remind you at this stage that the intensity of the colour depends on the concentration of sugar present in a sample. The darker the colour, the more sugar is present. We can usually carry out something called colorimetric analysis, which allows us to quantify the sugar present. I will cover this in another video, so look out for that very soon. The next biochemical food test I want to talk about is the emulsion test, which is used to test for lipids. We would add a small amount of the sample into a test tube and add ethanol to it and shake it. We then add deionized water to the test tube and should be able to observe any colour changes. If the solution contains lipids, we would observe a white or milky emulsion. You can see an example of the positive test in the photograph on the bottom right. Mark schemes do not accept a description of the emulsion as cloudy, so please avoid that in exam answers. The last biochemical test you need to know is the Biuret test for proteins. This uses a solution called Biuret solution, which contains sodium hydroxide and copper 2 sulfate, and it's blue in colour. You would add a small amount of your solution into a test tube and add the Biuret solution, and if you observed a lilac colour change from blue, then you would have a positive result. No purple or no lilac colour denotes a negative result, and you can see the photograph on the bottom right that shows the type of colour change that you would observe. Before we move on to exam questions, you should be aware that you could get asked to state how you would carry out a biochemical food test on a piece of food such as a muffin or seeds. The key thing here is that you must state that you would need to blend the solid form of the food and mix it with water to form a homogeneous mixture before you carry out the test. So, if we look at a few exam questions to give you some practice, the first question is asking you to describe how you would carry out the test for a lipid on a liquid sample, and this question offers 2 marks. The first marking point would be to state that you would mix the sample with ethanol and then with water. The order for these is really important. If you really cannot recall that ethanol is first, perhaps think of it as E for ethanol comes before W for water in the alphabet. The second marking point is for the colour change, so you would need to state that you would observe a white or milky emulsion. The second part of the question here asks you to describe a test for starch for 2 marks. The first marking point would be to state that you would add iodine solution. You could even say that you would add potassium iodide solution. The second marking point would be for the colour, so we just need to state that there would be a blue-black colour change where starch was present. The next question asks about carrying out a test to show that lactose is a reducing sugar, so you're expected to know that you would carry out a Benedict's test. This question is for 2 marks, so the first mark would be awarded for saying that you would heat the sample with Benedict's. Note that heat is underlined, so that is a really key point that you need to mention. The second mark would always be for the colour change, so we simply state that you would observe a brick red colour change if the sugar was present. Note that the mark scheme does accept the range of colours for Benedict, as it of course depends on the amount of sugar present in the solution. The next question states that lactase is an enzyme and lactose is a sugar, and asks you to describe how you would use the Biuret test to distinguish between the two of them. By now, you should know that enzymes are proteins, and this question is just for 1 mark, so they would not give you any marks for saying that you would add Biuret solution into both of the solutions. For the 1 mark, they actually want you to state the colour change for the enzyme, so you would have to state that the enzyme, or lactase, will give you a lilac colour. You could say that lactose will remain blue. The second part of this task asks you to explain why you would get that particular result. So all you would need to state is that lactase is an enzyme and enzymes are proteins. I hope this video helps you guys revise for the biochemical food tests. If you've got any questions, then feel free to post below this video. Please also like and subscribe to my channel for more content like this. Thank you so much for watching. Bye for now.